Thanks, Kyle. Thanks. Well, that was a little bit hyperbolic, I think. But you guys all know Kyle. I'm not a Hertz fellow, obviously. Yeah, I don't know Alex. I mean, I met him once, but. So this session is the biosecurity session, and I wouldn't necessarily call myself an expert in biosecurity, but I do work on emerging infectious diseases, and through my work in biosafety level 4 labs, of course, you have to learn a lot about biosecurity. So this intro might be a little ectopic as far as what we might discuss in the future, but I thought I'd give you guys a taste of what it's like to work in a BSL-4, a little intro to the pathogens we work with, and then at the end, I'll have a little brief introduction into bats and why they're reservoirs for some of these highly pathogenic viruses. So labs, so microbiology labs are divided into 4 different categories. There's biosafety level 1 through 4, and that's according to the risk for the pathogens or viruses that you work in those labs. So this also applies to bacteria and toxins, but today I'll just talk about viruses. So at a normal university lab, most of that is BSL-2 work, and you work with many, many common viruses such as seasonal flu, SARS coronavirus 2, hepatitis viruses, any of these viruses that you can think of that are low pathogenicity. Then you step up to biosafety level 3, and the labs have more infrastructure and more safety controls, and you work on things like yellow fever, West Nile, some of the more pathogenic flu viruses, SARS-1 and MERS coronavirus, and a whole host of other viruses. Then you step up into biosafety level 4 with a lot more engineering controls, and we work on more deadly pathogens, including Ebola virus, Marburg virus. So I highlighted Marburg virus here. You can kind of keep that in the back of your mind because I'm going to speak a lot about Marburg virus in the next 20 minutes. Lassa virus, Nipah, a whole host of other viruses. I have smallpox here in italics. It's a BSL-4 virus, but it's a little different category, so I'm not gonna mention smallpox. That's kind of ectopic to these other viruses. So why are these viruses classified in these different biosafety levels and risk group levels? So there's really 3 factors that you kind of blend together to decide if your virus is BSL-1, 2, 3, or 4. And those are the disease severity, so how severe is the disease if a human is infected, the transmission potential, especially in a laboratory setting, and the availability of treatments or vaccines. So you could have a highly pathogenic virus, but if there's a treatment or vaccine, that could bring it down to a lower biosafety level. So today I'm just gonna talk about biosafety level 4 and risk group 4 viruses. This is a picture of our lab in Berlin at the Robert Koch Institute, where I'm a group leader. And you can see this is a suit lab, so we wear positive pressure suits that are connected to air, and the whole lab is under negative pressure to prevent escapes of viruses. So we have 4 BSL-4 labs in Germany, and you guys here in the United States have 9 operational biosafety level 4 labs. So instead of droning on and on about the lab and what it's like to work in there, our institute has put out some public PR campaigns to show people what it's like to work in the BSL-4, so it's a good way to keep in contact with the local community so they know what we're doing in there. So I'm just gonna play 3 short, about 1-minute videos to get a sense of what it's like to work in a BSL-4. So this first video is just showing the suit room. So these are our positive pressure suits. And we have our biosafety officer, Frank, coming into the room. So first you don scrubs. You walk through a personal shower, which you don't take a shower. It just acts as an airlock. Frank's gonna do a suit check. So we check our suits once per week, and we change the outer gloves. And we make sure the suits are airtight. Of course, the most important parts are the gloves and the face shield to prevent infection. And then after the suit check, you wear a second pair of gloves over your primary gloves. So you have the suit gloves and 2 pair of gloves under that. Some labs use radio communication, so you can speak to each other in the lab. And then he's gonna don the suit, which is kind of awkward to get into. And he's ready to go in. So you have to connect to breathing air, and the suits are very big and oversized, and that's your reserve air. So as you walk from room to room, or biosafety cabinet to biosafety cabinet, you unplug from air, and you have approximately 5 minutes of reserve air time in your suit, and by that time, it's highly recommended that you plug back into air. I think there's been no one that hasn't plugged back into air. We read about that. So this next little video is actually the head of the lab demonstrating what it's like to work in the BSL-4. So all of our viruses are stored in a central repository in liquid nitrogen tanks, which has an extra access control, so only a few of us can get in there to pick out the viruses that we want to work with. And then we have what looks, if you didn't have the engineering controls, it looks like a normal BSL-2 lab, where you have biosafety cabinets, incubators, microscopes, centrifuges, and some other equipment, and a few pieces of specialized equipment depending on the lab. So all the sample work with live infectious virus, we work in biosafety cabinets, and that's to protect ourselves, so another layer of protection, and also to protect our samples from contamination, just like in a normal BSL-2. So we have very strict rules, obviously, for disinfection. You're only allowed to spend 5 hours in the lab, and then you have to come out and take a break. And we have a very strict buddy system, where someone either has to be in the lab with you, or someone has to be ready to go into the lab at a moment's notice. And then we have a two-way communication if we have an emergency and somebody has to come into the lab. And some weird stuff like oversized keyboards because it's really hard to type, and 3 pair of gloves, loudspeaker phones because you can't really hear through the suit very well. So this next video is just how you exit the lab. So as you enter the lab, you pass through a chemical shower that doesn't activate, but it acts as an airlock. And then when you need to exit the lab, you go back through that shower that activates. So first you dunk your hands to make sure there's nothing on your hands and you don't have a hole in your gloves, which actually is scary, a little common actually. And then you take a 3-minute decontamination shower, and this is a chemical called microchem that neutralizes membranous or viruses with envelopes. Of course, clean your boots. This is by far the most boring part of the day. The water's cold. You don't have those amazing epiphanies that you have in a normal warm shower. Typically, you have to pee if you're at your 5-hour limit. And then you hang your suit. Then you take your scrubs off, so you doff your scrubs, you take a personal shower, and then you put your street clothes back on, and then you go to the restroom. So an entry and an exit, just that transit time takes about 25 minutes, plus the decontamination of a hood takes another 5 minutes. So even to run in to do 5 minutes of work, it takes about 40 minutes of your day. That's a little cumbersome. So I've worked in 3 BSL-4s. I did my postdoc at the NIH in the Rocky Mountain Lab BSL-4 that just went hot when I arrived. I'm also a guest researcher currently at the CDC in Atlanta, where I've been a guest researcher for the last 10 years. And for the last 7 years, I worked at the Robert Koch Institute. And our BSL-4 is that white building that you can see within the brick building here. We also built a makeshift BSL-4 in New Mexico where I did my PhD, and there we could use the local fauna, so deer mice infected with hantavirus. I'm just curious, how many people have heard of hantaviruses? Okay, yeah, it's pretty popular in the US. I'm always surprised how many even non-virologists know about hantaviruses, especially when I'm out climbing and I talk to other climbers or hikers. It seems like everybody knows what hantaviruses are. So as Kyle mentioned in his hyperbolic introduction, I also attended, or I went to the Ebola outbreak for a few sessions, and there we set up a makeshift BSL-4 so we could do some research and sample processing. But that lab unfortunately had a very poor HVAC system, unlike our current labs, but we all came back healthy. So a little bit about the viruses. There are 6 major families of risk group 4 viruses. And of course, the most famous is Ebola virus. I think, show of hands of Ebola virus? Okay, good. Marburg virus? Okay, we're gonna do very well, I think, in this crowd. So those, of course, are the most famous. But the most deadly, as far as number of cases and number of deaths, is Lassa virus, where 5,000 to 10,000 people annually die, unfortunately, of Lassa fever. What's considered potentially on the high list of pandemic-causing viruses for the risk group 4 viruses is Nipah virus. Have people heard of Nipah virus? That's carried in bats, and that was the major protagonist in the movie Contagion, if you've seen Contagion. That's actually a fairly good movie, and it's, I think, Kyle's favorite movie, he told me. Yep. Kyle loves viruses, but he's a stem cell biologist, unfortunately. The US resident, we already went over this, is hantaviruses. So actually, in tissue culture, in a petri dish, these are BSL-3 plus viruses, but as soon as you put it into an animal, it becomes a BSL-4 virus. Crimean Congo hemorrhagic fever virus, show of hands. No, that's why I have most under the radar. Actually, this is expanding greatly, and it causes, I think, more deaths probably than any other virus except Lassa virus. It's spread by hyaloma ticks, and as the ticks are expanding, we have expanding range of host or human disease. And actually on Tuesday, when I was sitting in the airport, I saw there was another case in Greece where a 72-year-old gentleman died, unfortunately. And then we have the tick-borne flaviviruses. This is a special case because there's actually a vaccine in Europe, but we don't have that vaccine here. So for these tick-borne encephalitis viruses, they're BSL-4 in the United States, but they're BSL-3 viruses in Europe. So I know you probably can't read this. Maybe you can. This looks better than I thought. But I just want you to get the gist of this slide, and this is emerging viruses or expanding geography of some of these viruses. So since 2003 to now, we've had outbreaks or infections associated with new viruses, and I've starred the risk group 4 viruses. So this is a continuous discovery of pathogenic viruses, and some of these are risk group 4. And what's even more alarming, like I mentioned in the last slide, are things like Crimean Congo hemorrhagic fever virus, Marburg virus, and Lassa virus are greatly expanding in their geography. And actually, I came across this slide about a month ago, and it's actually even alarming to me. So dig a little deeper into these viruses. All these risk group 4 viruses are RNA viruses, which allows them to mutate and host switch fairly rapidly. They're enveloped, so this is a picture of Marburg virus budding from an African green monkey cell in a petri dish, and as they bud, they steal a little bit of envelope. They're very simple, but economical, so they only have between 4 and 10 genes and 10,000 to 20,000 bases. So if you remember Thursday night's talk, the P53 gene is 20,000 bases, so that one gene is the same size as the whole genome of several of these viruses. And this is just how, on the bottom, how we represent the genome, where we have, this is the genome of Marburg virus here, and it only has 7 proteins, but these proteins have many functions. So we don't fully understand how these viruses replicate and do all the things that they have to do, and how they cause disease. But there are several steps, and they're highly regulated. So first, they have to form these stable structures. They have to be able to transmit efficiently outside of a cell, and then enter very specific cells. And then in the cell, they have to hijack the host machinery because they don't have the ability to replicate themselves. Then there's a very complex budding and assembly process where they bud through the membrane, and they have to do that over and over, all while evading the immune system. So to do this, all of these proteins, so there's between 4 and 10 proteins, these proteins have to have many functions. And a lot of these functions are known, but I think there are several functions that we haven't described yet. And these proteins either act together, so the viral proteins acting with each other, for example, to form a polymerase, or they interact with host proteins to do things like inhibit the immune response. And I think of these proteins as like multi-tools. And if you mutate a little bit of this tool, the whole thing's gonna fall apart. So these are very economical and very specific for these proteins. So even though they're RNA viruses and they can mutate very rapidly, they're really locked into the constraints of the ability to do their functions. So all these risk group 4 viruses also are born from animals, so they're zoonotic. And in the animals, they circulate without causing disease. So for example, in bats and rodents, which are the biggest hosts of most of these risk group 4 viruses, the viruses happily replicate and stay maintained in the population of whatever animal is the host. And then it's when they transmit to humans, either directly or through an intermediate host, that we have massive disease. So this is a model that I work on, and a good example of this. So Marburg virus is hosted in the Egyptian fruit bat, where it replicates very well. The bats seem to be able to control it after a few weeks, but it's still maintained in the greater population of bats. And as soon as Marburg spills over into a human, it's up to 80% lethal. So there's a lot of challenges and a lot of things we still don't understand about these viruses, like what are the mechanisms that they cause human disease, and how are these animal reservoirs able to avoid this disease. For some of these viruses, we don't even know the reservoir. So Ebola virus was discovered 49 years ago, and we haven't found the reservoir. We think it's bats probably, but no one's been able to identify or isolate virus from a bat. We don't know what drives spillover. We have correlates of spillover like forest fragmentation, climate changes, seasonality, food scarcity, things like that, but we don't really know the mechanisms. And of course, the big thing is, can you develop a treatment or vaccine? So I work a lot with bats in our BSL-4s, and bats are really unique and special in that they host a lot of viruses, so their virome is very big. But there's also 1,400 species of bats, which is 20% of all mammalian species, so maybe it's not surprising that they do host a few of these viruses. But there are some unique features of bats that might make them good hosts. Things like powered flight, so there's this flight equals fever hypothesis, that during flight their body temperature rises and they have this cytokine response that might be able to control infection. A lot of bats hibernate, and that's another way to regulate their metabolism, and maybe that helps them live with these viruses. There's a lot of differences in their immune system. Our most recent common ancestor is about 70 to 90 million years ago. So of course there's a lot of divergence in the immune system. It's thought that these bats don't really have much of an inflammatory response. So that might be a way that they can control infection. And of course they're very ecologically diverse. Some bats migrate great distances. There's bats that are frugivorous, insectivorous, pescivores, so only fish-eating bats, which is really cool. They're actually echolocators. There's bats that eat frogs, so there's frog-eating bats. So there's a great diversity in bat ecology that might lend them to be able to either contact and host these viruses or transmit these viruses. So we work with bats in our BSL-4s. So on the left, you can see our lab in Berlin. So we go to Africa and trap various bat species. And lately, we've been working on Angolan free-tailed bats, which we think might be a reservoir for Ebola virus. So we trap them and quarantine them. These are insectivorous, so we have to train them to eat mealworms because we don't want insects flying around in our BSL-4. And we bring them back, infect them with viruses, and do some virology and immunology with those bats. Then I mentioned I'm a guest researcher at the CDC as well, and at the CDC we have a colony of Egyptian fruit bats, which are the natural reservoir of Marburg virus. So there's a lot of myths and a lot of speculation about these bats, I think, even in my field. And one of the myths is that all these bats carry all these dirty viruses. The bat virome is very big. There's thousands of viruses in bats, but there's 1,400 bat species. In reality, it's very tightly regulated in a tight co-adaptation that only specific viruses can survive in specific bats. So we tested that in our BSL-4s. We brought our Angolan free-tailed bats into our lab and we infected them. We tried to infect them with Marburg or Ebola virus. And only Ebola virus replicates well and is shed. And then conversely, for the Egyptian fruit bats, which we know are the reservoir of Marburg virus, we can infect them with various viruses, and only Marburg virus grows. So it's not like these bats are flying around like sponges for any virus, and they're great at hosting viruses. It's a very specific co-evolved situation. So my interest is bat immunology and how they are able to respond to these viruses appropriately. But our field is very crude in that we don't have a lot of reagents to study bats because they're all outbred. It's not like a mouse where you can just take a mouse off the shelf and order any reagent you want. So we're doing things like trying to find antibodies that cross-react. We infect bats with viruses and look at transcriptomic changes. We try and do antibody assays. But we're at the very early stages of bat immunology. So I'm going to show you just a tiny bit of data. One piece of data is comparing the bat transcriptional responses in the blood to primate transcriptional responses, and primates are a model of human disease. And we also actually have some human samples, unfortunately. Fortunately and unfortunately. And then another thing I've been doing in the last couple of years is trying to play with the immune system of the bats using drugs to turn down their inflammatory response or turn up their inflammatory response to see how they're responding and to try and understand how they're living with these viruses. So this is the big scary data slide that's actually very simple, I think, for you guys, because you guys are super smart. So on the left, we have the transcriptional responses of the Rosetta's bats infected with Marburg virus. And you can see nothing's really happening. So this is this lane here, and these are just different time points. So we infect the bats and bleed them every day. And on the right, we have the non-human primate and human data. So each column is a different study separated by the gray bars. And then each row are different genes, so we can look at the transcriptional responses. And what's very obvious is that the non-human primates are responding, and most of the genes that are coming up are involved in inflammation. So they're having this massive inflammatory response, but there's also downregulation of very specific genes, and a lot of these genes are involved in the adaptive immune response. So things like CD3, which are markers of T cells, and CD19, which are markers of B cells, those are greatly downregulated. And we can look at this in the blood. We can take a sample from the monkey blood after we've infected them, and we can see a decrease in the lymphocyte loads. So we see this dysregulation that's not happening in the bats. The bats are responding a little bit. There are other genes that are going up and down, but nothing drastic and nothing involved in these inflammatory responses. So Marburg virus, the results, virus replicates, but they have some appropriate immune response somehow. But in humans and non-human primates, the viruses also replicate to higher levels, and you get this disparate response. You have hyperinflammation, but also impairment of the immune system. So lately I've been trying to model that in the bats, and to do that, I try and mimic this immune response by impairing the immune response in the bats using immunosuppressant drugs, either cyclophosphamide or dexamethasone. We look at the outcome. So these are very simple experiments. We take bats into the BSL-4 and infect them with Marburg virus. In this experiment, we use dexamethasone to inhibit their immune response. Dexamethasone is a common drug, so if you need an organ transplant, you'll probably be put on dexamethasone or prednisone or a similar drug. So that decreases your immune response, so you don't have a response against the tissue engraftment or the organ engraftment. And the results were very striking. So we could inhibit the T cell and B cell responses in these animals. And if these bats had Marburg alone, they looked very healthy. I think everybody's finished breakfast here. So on the left, I should have said that before so you could look away, but I don't hear any forks or cups. So this is what a nice normal liver looks like in a healthy bat, even though it has Marburg virus. And on the right, you can see actually two of our bats. One was unfortunately found dead in the cage 12 days after infection. And this bat here was about to probably succumb to Marburg virus disease on that same day 12. And we can take cross sections of the tissues and look for viral antigens, so looking for viral proteins. And you can't really see it very well in this graph or in this slide, but there is some pink here. So Marburg virus is replicating to very low levels. But on the right, you can see massive increases in Marburg virus staining. So these are Marburg virus proteins. So in conclusion for that, the bats aren't just sitting there tolerant and happy to have this viral infection. They actually have to have their immune response fight off this infection, or they die of Marburg virus disease. So we also want to correlate this with potential spillover. We don't know much about spillover of Marburg from Egyptian fruit bats to humans. or non-human primates. Actually, in the wild, non-human primates are infected as well. But we think that by modeling the stresses that these bats have in the natural environment, so stresses like pregnancy or food scarcity, which are mimicked by dexamethasone, that might be a drive over spillover, or a drive of spillover. And we actually see that. So if we treat these bats with dexamethasone and swab them, we have a lot more virus coming out of the bats that are immunosuppressed. So this weakened immune system might be a correlate of spillover from these bats. So just to summarize, you can read this as I'm talking. Our humor in the BSL-4 is not that sophisticated. These risk group viruses are highly pathogenic and spill over from animals, so they're all animal-borne. Someone just finished reading it. We're finding new risk group viruses all the time, and they're expanding in geography actually very rapidly. In the last 4 years, we had 4 new countries have Marburg virus outbreaks that had never seen Marburg virus before. And the big mystery is, how do these bats survive infection, but humans have this massive response that results in disease. And we see that Marburg virus induces this extreme dysregulation. We don't know the mechanisms yet, but this dysregulation somehow doesn't happen in bats. And bats are actively fighting this infection, we were able to eventually show. And we hope that somehow this will lead to therapeutics. Can we train the human immune system to look more like a bat immune system and avoid this hyperinflammatory disease? So thank you for your time. I didn't show a lot of data, but all of the work was either done at my lab in Berlin or my trips to the CDC at the Viral Special Pathogens branch, working with John Towner there. So, yes, thank you. And I think thank you very much.